Protein molecules lie at the heart of biology. Our typical understanding of proteins states that each type of protein has a specific three-dimensional shape that enables it to perform its function. This dogma is challenged by intrinsically disordered proteins, which makes up one-third of all proteins and have central biological functions even top their shapes are constantly changing. Until now, our understanding of the structural properties of this stringing class of proteins has been based on studies of only a small number of examples. In research published today in the journal Nature, researchers from the Department of Biology, University of Copenhagen have shown how all approximately 28,000 disordered proteins in the human body behave. I have always been fascinated by intrinsically disordered proteins because they seem to defy most of the rules of how a protein should behave. For the last 20 years, we have worked on figuring out how these strange proteins look and whether new rules need to be applied to describe them. For the first time, we have now been able to study the structures of all human disordered proteins and begin to provide order into this world of molecular disorder, says Professor Christian Linderoth Larsen, director of the NNF Center PRISM, in which the research was performed. The goal of the PRISM Center is to combine computational methods from biophysics and machine learning with methods from cell biology to study how genetic variants cause disease. But until now, the researchers did not know how most disordered proteins look and hence could not even begin to study how mutation in the genes encoding for them might be able to cause disease.